Hey everyone, thanks so much for being here today for our last armor seminar of the spring series. My name is Matt Nelson, I'm the chair of the seminar committee. And before we get started, I just wanted to thank a number of people who were extremely instrumental in making this seminar series possible and really wanted to recognize them. First off is Jingmei O'Connor, Sarah Ruan, Stacy Drake, Abigail Derby Lewis, who are all part of the seminar committee as well and who have done a really thoughtful job of bringing in really exciting speakers to be here. There's a lot of work that they've put in that we don't necessarily see on the outside end, but behind the scenes, there's a ton that they've been doing. Also want to thank a number of the administrators who've really done the heavy lifting here. Amanda Dick, who for instance made cakes today, if you haven't noticed. Jonathan Hentz, Emily Halleck, and Teresa Riley. They really are very integral in providing a lot of the logistical support, of which there is very much involved in this process. And then finally, I just want to recognize the IT team and thank them for all of their hard work. Kevin Lewandowski, Tim Scare, Jason Gooseman, and Aurelius Barnwell. They've really done a lot to help make it possible so that we're able to do these Zoom seminars. And finally, I just want to thank all of our speakers and all of you who've been coming here to see these seminars, both in person and on Zoom. And I want to say, we're really excited about the fall. We'll be back then with a number of other seminars. And so yeah, I'll turn it over to Stacy, who's going to provide an introduction. And thanks a lot, everyone.
Hi, everybody. Thank you all so much for joining us for another ARMA lecture, the last of this season. I'm Stacy Drake. I'm a collections manager in anthropology, and this is the second lecture of May where we've partnered. This is hosted in partnership with the museum's inaugural celebration of Asian American, Native Hawaiian, and Pacific Islander Heritage Fund. So I hope that you all have been able to enjoy some of that programming throughout the month of May. Today, I have the honor of introducing one of our newer members to the Fields Collection Center team, Dr. Rachunliu Kamei. Did you okay? Yep. Chun is a Naga of the Rongmai tribe from the northeastern region of India. She received her PhD from the University of Delhi and subsequently obtained a Marie Curie postdoc fellowship at the Natural History Museum, London, in which she focused on the then understudied caecilians, which we will be hearing more about today. And these are from the poorly explored northeastern region of India. Her field-based research on caecilians resulted in the most comprehensive study of these creatures from any one region in Asia. Her primary research interests are taxonomy, systematics, biogeography, and the natural history of amphibians, reptiles, and swamp eels in India. Chun has published much of her research, including the discovery and description of many new species of caecilians, frogs, lizards, and swamp eels, and a new genera and new family of amphibians. I am realizing how underaccomplished I am, as well as rediscoveries of species long thought to have been extinct. Chun is also committed to public outreach, particularly to the natives in rural parts of the northeastern region of India. Today, Chun will be presenting a summary of her fascinating research on caecilians and amphibians, the world's least studied group of vertebrates. She will also discuss her professional journey that has brought her to her current position here at the Field Museum as the collections manager of Amphibians and Reptiles Division at the Field. So without further ado, welcome Chun. Let me go back to the, okay. Kevin? Yeah, we got this. Oh, can you mute me? Yeah.
All right.
Okay. Good afternoon, everyone. Thank you, Stacey, for reaching out to me to give this talk and for your effort to give an opportunity to a staff member in the GANS
Family Collection Center to highlight the research. I am deeply honored to stand here today in front of you. My introduction is going to be a bit long. Foremost, I want to dedicate my talk to the people in Manipur, my parents' native place, where a lot of families are, as I stand and speak here, suffering from violence, and many families have lost everything and fleeing their homes for safety. In joining the US celebrating Asian and other culture this month, and in honoring my family and my tradition, especially my deceased father and my 82-year-old mother, I'm wearing my traditional jewelry. My parents worked very hard to promote, protect, and conserve our tribal identity, culture, language, and ethics. And I considered wearing my sarong, but in the last 2.5 years, I've lived in 4 different countries in 3 continents, and I don't know where my sarong is. So I have to just make do with these necklaces. So although some of you may have heard from the introduction from Stacy that I belong to an ethnic group called Naga, I'm sure none of you have heard my tribe's name ever. I'm from my Naga tribe from Northeast India, and I've been asked to share about my journey. That's the tricky part, but I will try. And so I was 8 weeks old here when Stacy reached out to me to give this talk as part of the Asian American Native Hawaiian and Pacific Islander Heritage Month. And I freaked out mostly because I am not an American. And according to my visa category and in the legal parlance of the US government, I am a foreigner called a non-resident alien. And every time I move residence, before and after the move, I have to inform the lawyers and the HR and file what is called the change of address, aliens change of address card. So, and apart from imposing as an American, I was also most concerned about taking someone else's spotlight. And I hope that I am not offending any colleagues from the appropriate heritage group by standing in your place today. So let's dig in. Welcome to the Caecilians cake. First off, Caecilians are amphibians. The word caecilian invokes a variety of human emotions. The word, some of them they have never heard. Some, like a lot of my family, are astonished why on earth I would like to spend my prime lifetime digging around like a farmer, sunburned, emaciated, stunningly slowly, swell-dwelling creatures, wanting to become a doctor of them when there's not enough doctor for human beings. Some just don't care. Some are disgusted, like a forest department officer asked me if their ugliness and sliminess could be due to a bad karma in their previous life. Some know about them, some know about them and they shout, I love Caecilians, they're amazing, and I love those people especially. And some are simply terrified because of their snake-like appearance and movement. Scientists classify biodiversity. Okay, the Zoom people can't see the laser pointer, and I taught in college for 6 years, and I like to point out stuff. So scientists classify biological diversity in the scheme of taxonomic classification. Species is the smallest biological unit, and similar species are grouped in genus, and similar genera are placed in family and order and class and so on. And the class amphibian has 3 orders. The Anura, which includes the famous frogs and toads that you're all aware of, Chordata, that is salamanders and newts, and the special Gymnophiana, a distinct class for Caecilians. Now, a single line description for Caecilians they are tube-like, legless, snake or worm-like ringed amphibians of the old and new world tropics. To date, there are only 221 species of Caecilians known globally in 32 genera and 10 families. Now, one of the most hotly debated topics in the evolution of origin of extant and modern amphibians, called the Lysamphibia, is the order. One of the hottest debate in vertebrate evolution is the origin of modern amphibians, called the Lysamphibia, and there are many hypotheses, but the 2 main competing hypotheses are the Procera and the Batrachia hypothesis. Now, Caecilians properly separated from the lineage comprising the frogs and salamanders group about 300 million years ago. Caecilians are Gondwanan in their origin. Some taxa dispersed out of India into Southeast Asia, that is Laurasia, probably during the Oligocene. Now, long before, excuse me, I have something in my throat. By the way, Kate Columbus, are you here? I made the slide for you especially. So long before salamanders and frogs came about, and millions of years before dinosaurs roamed the earth, is there a lot of disturbance?
There is. If you pull the mic away from your baseline a little bit, it should be okay.
A bit like this?
Yeah.
Is that good? So long before frogs and salamanders came about, and millions of years before dinosaurs roamed this earth, the ancestors of caecilians have been around, and they are an ancient lineage, which means they carry a ton of evolutionary information. And caecilians may be the most interesting by virtue of their phylogenetic relationships and are central to understanding the the history of early terrestrial vertebrate life. Although now the modern amphibians are completely limbless, their ancestors have limbs, and caecilian fossils are rare, and there's just about 10 of them as far as I know. And this year, researchers discovered the world's oldest fossil caecilian, and they called it a funky worm. And it was discovered just this year in January 2023. Now, caecilians are relatively rare, and field biologists who study them are rarer. There are only a handful of professional diggers in the entire world, and here's 5 of us diggers: Mark, Dave, Simon from UK, and Ram from India. And caecilians are the world's most understudied vertebrate group, and among vertebrate biologists, they are best known as being poorly known. Now, most caecilians are burrowers, predominantly soil, but also burrow under organic debris, rotten logs, etc., and a few species are aquatic. Now, they have a very cryptic lifestyle and mostly hidden from view and in muddy burrows, and they are rarely encountered above the surface. And as a result, they are much less conspicuous components of tropical rainforest ecosystems, in addition to their relatively inaccessible distribution in the wet tropics and complete absence of representatives in North America or Europe. Now, the Gymnophiana, the technical name for the group is called Gymnophiana, and it means naked snakes. Early naturalists thought they were snakes, but because of their conspicuous lack of external scales that were visible, they were called naked snakes. But many caecilians have scales. Some species are aquatic, and they're called rubber eels, or sometimes they're called land sharks because of their sunken jaw and terrestrial lifestyle. Now, caecilians are not snakes. Snakes are reptiles. Reptiles have visible external scales, and their skin is dry. And snakes and other reptiles have what is called the amniotic eggs, that is the hard-shelled or leathery eggs for largely terrestrial lifestyle. And caecilians are not eels. Eels are fishes. And with a few exceptions, fishes are entirely aquatic, and fish have gills and lack lungs. Only some larval caecilians have gills, and adults have lungs. Caecilians are definitely not worms. Worms are invertebrates. Invertebrates don't have skeletons, caecilians do. And caecilians are just not tube-like structures. They have a lot of fascinating characters. And I'm going to use my stuff again. And as you can see here, they have rings, are called annuli, all along the body. They have a very strong skull. The skull is heavily fossified for compensating soil burrowing. They involve a head first digging style. And a lot of them have 4 rows of teeth. And if you split open one of the rings, you see scales of rows, and there's a single scale that's enlarged. And that flower-like thing here is the phalodium, also called the phallus. The caecilians are different from all other amphibians in males having these intromittent organ, which is reversible. It's basically the most posterior part of the gut or cloaca, which everts, and all caecilians practice internal fertilization. Now they have a rudimentary, their visual system is rudimentary, and they have very small eyes, rudimentary physiologically and morphologically, and they have the little eyes are either entirely, sometimes on this particular one, it's quite big, but they are buried hidden under skin or bones. Now, but nature and evolution has ways to compensate this poor vision for caecilians. They have this thing, a protrusible sensory organ, and as they move, you can see them I mean, it's protrusible, and they are believed to have a similar function as snakes' tongues. We employ different methodologies to study and characterize caecilians, and one of them is soft anatomy. For example, this is the soft anatomy of a caecilian phalloidium. If they are everted, we study them. If they are not everted, we make a mid-belly incision and to study the knobs and nodules. So these are like, for example, you can see a lot of differences. These are different populations. We study the ridges, like the knobs and stuff like that. And like, as you can see from here, this one looks quite different from this one. And this particular one always reminds me of Elvis, you know, the vintage microphone that he used to use. Don't you think? Now, for caecilians that do lay eggs, they do so in terrestrial nests rather than in water. And a typical caecilian mother lays her eggs like a string of pearls, like a pearl necklace, and then she rolls them up into a bunch, like a grape-like cluster, and fastens them to a small root or twigs. Now, for a comparatively small group of just about 200 species, caecilians exhibit an extraordinary diversity in reproductive modes. There are species that have the typical amphibian lifestyle, that is, they lay eggs, they have then they have the free-swimming larval stage, and then they undergo metamorphosis. And then there are egg layers, but which are direct developers. That is, when they hatch eggs, when the embryos hatch, they come out as miniature adults. And then there are species that are life bearers too. And there are species which exhibit unpleasant sounding maternal investment style called dermatophagy or uterotrophy, a literal translation of which is peeling and eating mother's skin or eating the mother's uterine lining. I can see your faces. Now, let me take you to the world of Sir David Attenborough through a BBC documentary about the discoveries of this freaky sounding dermatophagy by my colleagues and friends.
One great group of burrowing amphibians has lost its legs altogether. You might think that this was a giant earthworm, but if you picked it up, you would immediately realize it's got a strong, firm backbone. It's a caecilian. Caecilians are found in almost all rainforests, but they are seldom seen, for they spend nearly all their lives underground. The female, having produced her young, stays in her nest chamber to protect them. Caecilian eyes are rudimentary. They're covered in skin and scarcely function. In the darkness underground, however, the animals have no need. The young enthusiastically lick a secretion from a gland at the end of their mother's tail, and their constant hunger seems to be the factor that keeps this little blind family together. In a single week, the young, incredibly, increase their weight by 10 times, apparently just from drinking her secretion. But could that be their only food? As we filmed, one of the youngsters revealed a clue to their rapid growth. It yawned. It already had hooked teeth like a baby shark. It surely doesn't need these if it's going to do nothing but thinking. Could it be feeding on something else? A few hours later, our cameras, for the first time, revealed the answer. There was a sudden frenzy of activity. The babies started swarming all over their mother. They were tearing at her flanks, ripping off segments of her skin that proved to be full of fat. Turned out that she regrew her skin every 3 days to provide her young with another nourishing meal. Blind, elongated, and legless caecilians may be, but simple, inoffensive earthworms they are not.
So Sir David Attenborough is really big in Europe and Asia. I don't know how big he is here. And I was watching this scientific documentary with my husband and about like amphibians, and he said, and amphibians are frogs and toads and salamanders and newts, and he left out caecilians. I was so upset. Okay, back to the top. So two significant reasons why I decided to do study caecilians in Northeast India is because they are oh. What happened? Because globally there are poorly studied group of vertebrates, primarily because they are very difficult to sample and they are rarely encountered in general herpetological surveys, and also because of their restriction to relatively inaccessible wet tropics of the world. And another reason is that Northeast India was a biological paradise, unexplored. Systematic and dedicated surveys have not been previously done. So India is a very big country, as you might know. And India is part of 3 global biodiversity hotspots. There are 36 hotspots in the world, and these hotspots represent only 2.5% of entire Earth's land surfaces. But nearly half of the world's plants and animal species and endemics are found in these biological hotspots. And endemics are those species which are restricted to one particular geographical area and not found anywhere else. And the phenomenon is called endemism. Now, the Northeast region of India is the easternmost part of the country, sticking out a bit like a chicken neck there. It comprises of, it shares the international borders with Bangladesh, Bhutan, China, Myanmar, and Nepal. And the region comprises of 8 states, that is Arunachal Pradesh, Assam, Manipur, Meghalaya, Mizoram, Nagaland, Sikkim, and Tripura. The Northeast region of India, as you can see here, straddles 2 biodiversity hotspots, the Himalayas and the Indo-Burmas. Northeast India till this, okay, Northeast India is still, to this day, quite underexplored. The region was traditionally called just a gateway or a dispersal route between India and Southeast Asia with strong South Asian elements. The region was thought to be a distinct biogeographic unit with its own ancient or special biota. And I didn't know about caecilians myself until I started a PhD. Now, when I started digging into the literature, I found that there are 2 amphibian hotspots in India, the Western Ghats, you might have heard, and Northeast India. So I compared these 2 the geographical areas and caecilian diversity and their endemism between these 2 hotspots in India. And I realized that Northeast India, which is almost twice as big as the Western Ghats, had only 4 species in 2 genera, while the Western Ghats had 24 species in 4 genera, representing about 80% of all of Indian caecilian and diversity. And so basically the Western Ghats took away all the caecilian glory in India. So both these hotspots have many similar environmental conditions, environmental and ecological conditions like they have a lot of rains, have rainforests, have many amphibian suitable habitats, and the frog diversity between these two biodiversity hotspots, between two hotspots in India, they were comparative back then. So the Western Ghats was comparatively much better studied than the Northeast India. So I decided to try and change that statistics by focusing on the Northeast region. And I found the first question for my PhD, if this perceived rarity of caecilians in Northeast India is true, and if there were really only 4 species from the entire region that spreads across more than 100,000 square miles. I had 4 extremely poorly known and poorly diagnosed species to start with. From the specimens I had in my, for me, was just about 10 to 16 museum samples known from less than 15 localities. So fieldwork was fundamental to my research. Field tool is very simple. A machete for clearing vegetation and a spade for digging. Now, with no external trace of their presence in their secretive habitat, finding caecilians requires a lot of back-breaking, exploratory digging. And this one was not staged. It was filmed as I was doing. They are slippery and wriggly. And if your hands are wet, they can wriggle through your fingers. And if you're unlucky to drop it in a grassy or leafy vegetation, it's just goodbye. Once we lost a specimen in open space and 3 of us were huffing and puffing, digging trenches and swearing and spent more than 2 hours. And by that time, actually, we didn't really want the specimen. It was just to soothe our bruised egos. Now, the field localities in these hot tropics, they are really hot. I'm not good with Fahrenheit, but like 36 degrees Celsius, that's about 100 degree Fahrenheit, you know, very high humidity. Drinking water is very scarce in these digging expeditions, and caecilians are difficult to find. So sometimes like you can gauge the level of frustration when you catch one and you lose it. So I dug in all kinds of habitats away from water, with water seepage, agricultural lands, forests, mountain streams, roadside ditches, alongside seasonal or perennial stream, one of the best spots. And in my earlier digging career, I didn't have wellies, gumboots as they call them here, and wore canvas shoes that never dry. So on several occasions I got trench foot, but when I got introduced to my British colleagues and friends, they brought me wellies from London. I also dug a lot around the edges of rice paddies, another good spot. And sometimes I get free help, like this little boy helping me to find caecilians. It's not child labor, by the way. I was digging in this paddy field, and the son of the village chief recognized me as the snake digger. And he told his boys that if they help me find a specimen that I'm looking for, the village will get a mention in local newspapers. So I got an army of little boys like going crazy. And I was getting concerned because caecilian digging requires some skills and a lot of patience that you just don't go around digging, you know. We also explored completely different habitats, like away from water sources, flipping rocks and boulders and plateaus and grasslands. And a lot of the digging days will continue after sunset. We got a cross section of caecilian occurrence across Northeast India, and these orange dots are some of the examples of localities surveyed. It's not a perfect map. In total, we put in more than 2,500 person-hours of soil digging surveys, sampled more than 80 populations from more than 300 localities, and we covered elevation ranging from 30 meters to 2,500 meters. And our effort was the world's most extensive soil digging survey back then in 2012. As part of sharing about my professional journey, let me digress from caecilians here a little bit. Alongside my PhD, I taught as a full-time assistant professor in St. Stephen's College, where I single-handedly built a lab for undergraduate biology course and started a new course. And alongside my teaching and doing my own caecilians PhD work, I played a primary role in establishing a fully functional natural history collections unit from scratch for my PhD lab, taking charge of specimen acquisition, processing, cataloging, databasing, etc., for the entire collection of Northeast India. And besides selecting survey localities, arranging permits, and all field logistics, because I was the first PhD student in my lab and also the only student from Northeast India. And during my PhD days, especially 2007 to 2011, when I did field work every monsoon, the lab acquired more than 2,500 specimens of reptiles, amphibians, and swamp eels. And I led field expedition in at least 16 out of 28 states of India. Now back to caecilians. So back from the field and after my teaching assignments and in the college and other PhD lab priorities, setting up the lab and everything, I would knuckle into specimen work, measuring hundreds of caecilian specimens. And now you can see here, I have a very sleepy head. Caecilian taxonomy is and can be complicated because they have very few external diagnostic morphological characters. Sorry. And caecilian taxonomy is not fully standardized. So our aim was to score as many visible characters as possible, and we were able to score about 50 characters per specimen. Like, for example, we count the number of rings on the underside, on the upper side of the body. We take a lot of linear measurements, distance between the eyes, distance between the eye and the tentacle, etc. And we split open and we count the number of scales, rows, at different sections of the body. And personally, for me, counting teeth in 4 rows, like really, really tiny teeth of 4 rows and under the microscope was the most difficult, like personally most challenging. It often took more than 1 hour for just one specimen. For characterization, we also use non-invasive techniques like radiography to count the number of vertebrae, which is a very important taxonomic character, and cranial CT scans. Skull characters are very important, especially for higher taxonomic groups like genus and families. And the plate on the right-hand side is a plate from an unpublished work. It's a bit too small for you all to see, but there's some difference going on between some bones here. I know you can't see it from there, but believe me, there are like some really cool differences in the bones. And of course, we use DNA to support our morphological and other data. My PhD was really like a jack of all trades. Since nothing was known about them, caecilians, I tried to study all aspects, including reproduction and brooding of one of the new caecilian species we described. I want to show here a mother with her clutch that I reared from early stage to post-hatching. And I just want to show you the level of maternal investment. Mothers brood for about 3 months, 90 days. And during these brooding, they eat minimally, at least in my experiments, shouldn't eat anything. And she constantly turned the eggs probably to provide to prevent infections, and they do this until they hatch. Now there are 2 families of caecilians that occur in Northeast India, the Ichthyophidae and Caecilidae, and these are the name-bearing types called holotypes in taxonomy of the 4 species from Northeast India. And as you can see, they are in quite poor states of preservations. One specimen is completely eviscerated, one is broken into 2, the head is just dangling off. And actually these specimens are not so old, but it was just not best conservation practices. And these 4 species were known either from a single species or a maximum of 3 specimens, and they were not sighted in life for decades. But my team were able to find new samples from the localities that were originally collected many years and decades ago. It just turned out that no one went to look for them. This is my first PhD publication, and here we describe the 3 new species of striped caecilians from Northeast India in the family Ichthyophidae. Now, this species called Giganophis fulleri was described in 1904 based on a single specimen that a British naturalist found in a bag of snakes that he purchased from the locals. And this species was not seen in life for more than 100 years and was thought to be locally extinct. But I dug them up in 2007. I was so thrilled. And this species was a biogeographic puzzle to caecilian biologists for a very long time. It was thought to belong to a Western Ghats genus Giganophis in the then family Caecilidae, and caecilian biologists thought that this species formed was at the easternmost limit of the genus distribution. Now, to resolve this biological anomaly, we compared the DNA, mitochondrial and nuclear DNA of our Northeast Indian samples, and the DNA of all other Sicilian lineages in the world and compared external morphology, especially skulls, and also estimated the divergence times, that is the ages of the different lineages. And our analysis showed that the Northeast Indian so-called Giganophis Sicilians, they not only formed a clade distinct from the Western Ghats, but were of an ancient lineage. And the split between the sister taxa, this is the time tree, and the split between the taxa took place about 140 million years ago, plus minus 20 million years. And our studies also revealed a surprising and unexpected phylogenetic relationship. The Northeast Indian Giganophis Sicilians were found to share closer affinities with an African exclusive family called the Herpelidae, rather than the true Giganophis Sicilians from Western Ghats. So, because of its distinctive morphology, inferred age, and phylogenetic relationship, we described the Northeast Indian Sicilian radiation as a new family, and we called it Caecilidae, using the local tribal Garo name Chicil for Sicilians. Now, describing a new family of vertebrates got the attention, a lot of attention of national and international media. And as a native of the region, I was glad the Northeast India was marked on the global Sicilian distribution map as a region of diversity and endemism. We made a YouTube video, and within a few months, we got more than 100,000 views, and Sicilians became very popular. And some newspapers called them frogs without legs. Yeah, and National Geographic selected our discovery as the third of top 10 scientific discoveries of the year 2012, even ahead of discoveries like lost world in Antarctica, sugar in space, a God particle, and one of our picture of a Chicala mother guarding her egg clutch was selected as the second out of 10 best news pictures of 2012. So all these was very interesting. These discoveries are interesting, incredibly exciting, but the ground reality is something else. There are threats, and the major ones are habitat destruction, human population increase, and pressure. The first and second go hand in hand, and specifically for Sicilians, probably the unfounded belief or misperception of Sicilians. Northeast India is an amazing place with a profusion of diverse habitats. We have rolling hills and mountains, but there are major environmental concerns and environmental and conservation concerns, and you see the landscape transforming where the lush green valleys and mountains are fading into burnt, barren lands. As you might know, India is the second most populated country in the world, and this is my hometown, bursting at its seams. And to feed the burgeoning human population, the government had very good intention, but they put up these arrogant and disturbing billboards to destroy our valleys and convert them into rice bowls. The region is being indiscriminately exploited to feed us human beings. And in my interviews, many interviews with the local people during my PhD surveys, I found that Sicilians are feared as fatally venomous, and they're called snakes all across Northeast India. And they have local nicknames like double-headed snakes or backache snakes, and old people in their 80s and young boys in their 5 and 6-year-old kids. They will tell me the oral tradition that if you see this animal, you chop them into pieces, flung them far, you do not touch them. If you're bitten, there is no cure. And the result? Persecution. There was a lot of intentional killing. Also Nagas, especially other natives in Northeast India are very well known for eating anything that moves. And in my observation, Nagas kill for 2 reasons. If they can eat them, they also kill them if they can't eat. And they say, oh, why do you kill? Say, oh, just time pass, you know? So there's no respect for a lower form of animals. So I committed myself to do a community education, but it was beyond the scope of my PhD. So I decided that I would do it after my PhD, because I know that knowledge can dispel fears. Now, if I had a live animal, I would show locals and sometimes ask them to hold and touch them. And on the upper left is a small village with no electricity, and the villagers called my presentation night cinemas. The turnout for the first night was disappointingly poor. I learned the following morning that the villagers did not turn up for my cinema because they were busy plotting to kidnap me with members of neighboring village because they were convinced that I was digging for gold in their village. Now, after the first day, when I showed them the live Sicilians and told them what I do, they said, "Madam, show us your cinema," but my cinema battery is gone, so I had to do on my laptop. And then the turnout, the entire village And even the co-conspirators from the neighboring village came to see my talk, and it was really like, it was a great moment. Now, typically, external speakers or visitors to villages or schools in remote places in Northeast India, they receive money from a bureaucrat or politician coming to give them speeches. So some of the places I was trying to talk or explore and give them a talk, they asked me how much they will get, how much I will pay them if they gave me audience, you know. So there's a lot of logistics and whatever challenges. And also in some places, okay. Sorry. So in a lot of places, I also asked them to repeat after me and to not forget what these animals are and how important they are, and that they should not kill them and that they are not venomous. Now, I directly addressed more than 6,000 varied members through some 30 popular talks. My audience was very varied widely, including bureaucrats, university students, researchers, but a great majority of them were school children and the non-literate farming communities who share a habitat with caecilians. It was a very special occasion for me as a homegrown and woman scientist to interact directly with the public, especially children, our future leaders. And kids in my area have never heard the word scientist except through textbook or TV, and they have never met a scientist, let alone someone who looked like them. So in some schools, I had really a rough time trying to shush them because the kids thought that I was the peon, like preparing for the scientists to walk in, you know. Actually, I don't work only on caecilians. I also work on other cool reptiles and amphibians. With my colleagues, I've published these several papers on frogs and geckos. These are some of the new species from Northeast India. They're all from fastly degrading and disappearing habitats. The site that I was digging in the earlier video is now gone. It's now converted to a fishery. Here's some more pictures that we discovered from Northeast India, and we still have many more to work on. I have also published on fish, and this is a thread-like swamp eel that I found in soil while digging for caecilians, and it was called hot pink eel, a catchy term that a reporter gave. And this hot pink eel became a media hit, and people said it looked like an alien, and the skull looks crazy, of course. And this species also got a comic strip by a guy called Green Humor. Now, this is another eel that was published this year with my colleagues, and so this is the summary of my research. Now, going on to the second part, my journey, like I said, the name of my ethnic group is Naga. Nagas are indigenous tribes, people inhabiting several Northeast Indian states and in Northeast India and across the border in Myanmar. When the British were leaving India in 1947, the Nagas expressed a desire to be a free people and not be part of Indian Union, but it was declined. So on the eve of India's Independence Day, we declared Naga Independence Day on the 14th of August. And we have our Naga federal government, our Naga flag and constitution. And the Nagas fight for right to self-determination has seen much bloodshed, and there are few families that have not suffered from the violence. And Naga insurgency is I believe, the longest running in South Asia. We're about 66 very colorful tribes, speaking about 89 different languages and dialects. My tribe has at least 3 distinct languages and dialects. Naga tribes share a lot of similarities in our traditional costumes, dance, songs, etc. But each tribe has their own distinct culture and costumes. For example, hornbill feather is very commonly used article. My Naga tribe's most popular dance is called the hornbill dance. But sadly, the hornbills are almost, if not completely extinct in Nagaland due to overhunting and lack of habitat. Rice is our staple food, and traditionally we make vegetable stews and meat. And rice is very important for Nagas, very important, it's our staple food. Pork is hands down the favorite meat, and especially pork fat, so much so that we ferment them to have constant supply throughout the year. And traditionally we eat food out of wooden plates carved out from a single wood with legs, hunkering down around fires. And I brought such samples of Naga plates with legs, and if you want to see them, you can come to my office. Nagas are musically very oriented. We're a very musically oriented and talented people. We're always singing or humming a tune while farming or weaving or just doing household chores. Here's a group of women singing a beautiful folk song while spinning yarn and weaving. These women are wearing blouses made of yarn from nettle plants. So the making of yarn from nettle plant is a special art which nearly went extinct with the infusion of modernity, but there are a few people now rigorously trying to bring back the art. Now, before exposure to and influence from the West, my ancestors were headhunters. American Baptist missionary brought Christianity to Nagaland in 1871. And with Christianity, we were taught that it is barbarous to cut off another human head and to take a human life. And eventually it was banned in 1960s. Okay, my transition is not working. So, and the man behind the tattooed, the man behind the tattooed man is smoking opium. Sharing opium around fires was a favorite pastime for warriors, and a session can last for 3 to 4 hours. Now, for a Naga warrior, I believe the proudest moment is when his face is getting tattooed by the wife of the village chief. And if a man has not taken heads, he only gets a tattoo on his face, on his chest, like this guy. Now, I love hanging out with older people from villages, chatting about Naga traditions, our ancestors, folklores, songs, etc. between different tribes. Here, I'm having a good time with Coco, a friend and a guide from a tribe bordering Myanmar, and a local expert on hornbills. My early childhood was in a thatch house, top left. That's my mom. She weaving a shawl. I wove many such shawls. My mother was a social activist and working for the upliftment of my tribe in particular, and Nagas in general, mediating between warring Naga factions. She has an indomitable spirit. She's a fireball. And my dad, a man of few words, determined and like water, and they were perfect for each other. My parents were very progressive in their outlook. During the time, my dad was thirsty for education, and in our traditional Naga culture, women could not inherit. Sons are a pride, daughters generally less valued. So my mom and dad sent us to the best local schools possible. They valued education and taught us that girls were no less than boys, and that girls can be stronger, more educated, more successful. We are 6 sisters. Each sibling takes care of the younger ones and pass down the trainings and life skills they receive from parents. And I don't have much memory about my childhood except studying very hard and weaving in candlelight in my spare time and singing and dancing with my sisters. Saturdays was handicraft time, shining pots and pans, cleaning the pigsty and collecting fodder for a week's supply for our Christmas pigs. And Sunday was rest, no work allowed. We get to watch He-Man, The Power of Grayskull, Tom and Jerry, Donald Duck just before going to church. This is a rare picture of me with my dad. I got the passion for community education from my father. I wanted to serve my community like my mom and dad did, but in a different capacity. My childhood dream was to become a medical doctor. I worked very hard all my young life to achieve that goal, and I wrote and topped the open exam among the Nagas in Nagaland state to get a tribal quota. There were only 2 seats to study medicine in India's top medical college, but when I was within the grasp of achieving my childhood dream, the opportunity was taken away from me. I'm a native, but due to intertribal political issues, I was rejected as my tribe was considered non-Indigenous. It still is. It got recognized recently, but quickly got de-recognized again. So at that young age of 15 or 16 years, seeing my name struck out as with a note that I'm rejected was very hurtful. I had to give up that dream, and the only career opportunities available to us then was either to become a doctor, engineer, or civil servant through open examination, through travel quotas. And my next real passion was PhD research, and although I got a PhD fellowship through an open national exam, I couldn't pursue due to family circumstances. So I tried to become a civil servant. So fast forwarding a few years, the state leadership seemed sympathetic towards a tribe, and I cleared 3 stages of competitive exams to become a bureaucrat. But when I went to collect my offer letter, the job was taken away from me again by a more powerful tribe. And back then in Nagaland, it was not enough to just take away your achievements. They would also vilify me in the local newspapers, and people from my tribe were not able to achieve much in that world. So I was yanked out of my comfort zone. I decided I will not rely on tribal quotas anymore and focused on doing my second original passion, that is research. Now, when I meet people from another community or my tribal political issues are discussed in various platforms, my example is always taken, and this issue comes up a lot every day. My example is taken and they recognize me as the one who was rejected twice. And another professor once described me as the Japanese golden joinery, that is broken pieces stuck together. And I thought it was a beautiful compliment. Now, financial emancipation was what I needed so that I can scout research opportunities and leave my family home. So in 2005, I became assistant professor in that college to be able to pursue my research dream and save some money for rainy days. But the PhD fellowship that I got had lapsed. And this was my CV. We call it biodata back then. I don't know why, biological data or something. So besides having a good academic track record, I had no idea, I had no publication. And my CV said things like, I got my first in javelin throw or shot put or discus, and I love knitting, and so this is where I started. And my bachelor's degree and master's degree was in botany, and with no idea and support how to do a PhD, I was like really lost, and I really struggled to find any botanist professor with resources or willingness to supervise me for a PhD in a rather inaccessible Northeast Indian biodiversity, because Northeast India is politically very unstable. Some states are very volatile. Civil infrastructures are lacking. And in 4 of the 7 Northeast Indian states, typical Indians require inner line permit because they are government of India's protected areas. And of course, field work is very important, is very expensive and very dangerous. But finally, I found a professor, but I had to switch from botany to zoology. My PhD took almost 8 years to complete. The results were fulfilling personally and scientifically. Our team's field-based research resulted in the comprehensive study of any caecilian from any one region in Asia. But it took a huge toll on me mentally, physically, and emotionally. And just before and after my PhD thesis submission, I lost 2 loved ones in my family, my father and my brother-in-law. In fact, my brother-in-law died on me when I was just 5 days away from finishing my thesis. Now, after I earned my PhD, I didn't have a postdoc planned out in advance, and I had resigned from my teaching assignment, and my pittance savings couldn't pay the bills and apartment and rent. So my international Skype boyfriend, now husband, recommended that I go backpacking. The thought was terrifying, but in a few days, I found myself frantically giving away all my belongings to charity, and then I hit the road, going to universities and institutions, giving talks on my own terms, networking, and writing 3 project proposals. And all those 3 project proposals got funded, and it was really motivating. And 2014 was my golden academic year. And I backpacked until I came to the Natural History Museum to start my Marie Curie postdoc. But one day I just lost steam. I became desperate to leave academia. I had 4 heroes, these 4 men. They said, take as long as you need, but don't leave. Well, they didn't actually say don't leave, but it was always implied, and they were always there for me. And the fellow on the right here, the Steve guy, is my long-term collaborator and my husband. We, the Nagas, have very intricate tradition for marriage. It's asking for a woman's hand for marriage is a multi-step tradition, and it goes over a prolonged period of time. A man has to pay customary bride price called a dowry, and paid in the form of pigs and rooster, livestock, land, jewelry, etc. So my mom asked my Steve for 2 piglets, and the role of a man in Naga tradition is men spearing the pigs and taking part in the butchering. But of course, he didn't have the stomach for it because he's a lad, a city boy from Dublin. So my family let him off the hook. And looking forward to the piglets cooked in Naga style. We got a blessing ceremony in family home. Modern Nagas wear the Western white flowing gowns, but I wore my traditional sarongs that my mother saved for me for years. Now that I was married, I didn't know what to do. I wanted to become a housewife, but I made a poor decision in marrying a very poor herpetologist. I saved a ton of money during my postdoc, but it was nothing for a life in London. So Steve also got a Marie Curie postdoc to do in NHM and in the Natural History Museum and University College Dublin. And he supported as long as I needed. And these were the options that I considered, and I seriously, briefly pursued some of them. And along with my guiding principles and 7 philosophies in life, I was a caregiver. I tried doing a child minding, copy editor, rock climbing. My husband said, "You're a bit too old now." So okay. And while pretending to be a fish digger, presenting to a housewife, I was also publishing, like for example, that hot pink eel, and this editor guy wanted to run a story, and we had a nice long interview, lunch, free lunch, and in the end he said, "Oh, so what are you doing now?" I said, "I'm retired, I'm a housewife," and showed my knitting that I took out from my bag, and I didn't know that he would publish that, but I have on record that I tried to be a housewife. Now, after not being so good at being a housewife, I had an epiphany one day that I would love to give back something to science, and the Caecilians in Northeast India, in the NHM London has still not got the attention of the relatively new curator, Jeff Stryker, so I volunteered to curate the Caecilian collection, and that's me and Jeff exchanging hats for a photo. He doesn't generally wear pink hats. So the actual number of Caecilians were unknown, and because specimens were registered as lots, and the taxonomy had not been updated since the 1900s for most of the collection, including the Caecilians. So I archived the original labels, issued barcoded label systems, georeferencing, extra, physically rearranging the collection based on current taxonomy. And our work demonstrated that the museum has, the collection was much richer than previously thought, both in numbers of specimens and number of taxa represented in the museum. And we also discovered a new genus that was previously unknown in the collection. So on my housewife budget, I went to present our work at the World Congress of Herpetology in New Zealand in 2020. Now, I had not taken any real holiday in my life, and so I plan to take at least two years off to rejuvenate myself. But after my postdoc, but the COVID-19 pandemic hit, and no one was hiring. By mid-2019, I put myself in the job market, freaking out. I applied for 6 jobs and applied for this position twice. And except for the one that was quickly rejected, I was shortlisted and interviewed for all the applications, and I began to know what I wanted to do or what I could do. And you know, life in academia can be very challenging. We feel burned out, we lose steam on our long journey. It takes a lot of toll on us. And if there's any friends and colleagues here in the audience today feeling the same way, I want to say, hey, you are not alone. Take a break if you must. Take as long as you need. Try and find a support group, but try and come back. You all know, but let me introduce briefly my colleagues in our division. Joshua Mata started as collection assistant about 5 years ago, and is now the assistant collection manager in our division. Josh did a fantastic job looking after the collection between my predecessor's retirement and my arrival, and Sarah Rowan's the assistant curator hiring in the field. Alan retired recently-ish, but colleagues from around the world are still emailing him, and he's still and helping researchers and guiding me when I have questions about our collections practices and history. And I want to make our division a badass department because it deserves to be. And Sarah said, "Come here, girl. We'll make our department badass." So the Amphibians and Reptiles collection ranks among, our collection ranks among the top dozen collection in the world and among the 6th largest collection in the United States of America. It is also one of the best maintained herpetology collection. We have more than 300,000 specimens, maybe representing about 6,300 species of reptiles and amphibians altogether. This number could be inaccurate, but I think a museum has at least 1/3 of all herpetology species in the world. We have one of the strongest collections from Southeast Asia. We have nearly 700 specimens of Caecilians representing 9 families. And I don't know yet, but I won't be surprised if our Caecilian collection is one of the best and most representative collection in the world, both taxonomically and geographically, but I don't know yet. I will find out. Now, our division went without a collector, without a curator for a very long time, I think for more than a decade, until Sarah was hired just over a year and a half ago. Not having a curator means lack of active field work and no new sample acquisition. Now, amphibians and reptiles are the world's most threatened vertebrates, with more than 40% of amphibians and 20% of reptiles threatened with extinction, according to 2020 IUCN report. Now, we all know that physical specimens represents priceless timestamps of our world biodiversity. Biodiversity is staring us in the face with climate change, habitat destruction, etc. And we are currently faced with a reality where ecosystems are just vanishing behind curtains for us to only realize afterwards that we never found what lived there or how many species we have lost. We need to actively document and curate samples if we are to understand what species still exist in their shrinking islands of natural habitat, if we have any chance to protect some of them. And I'm delighted to be part of the Field Museum team, to have the opportunity to contribute in some way to our field of study as custodians of this fine collection, and to represent our vulnerable and often underappreciated and misunderstood slimy and scaly friends. The anti-harassment statement, I believe, here in our forward-thinking and inclusive cultural and educational institutions, such as our institution, and I believe we follow this statement and commitment in letter and spirit. There are institutions in less economically developed nations where typical life in academia is not safe, and there are no safeguards to protect against bullies. Students commit suicide regularly in India. Just in the week of May 10th, a PhD student in my alma mater, Delhi University, allegedly committed suicide due to mental stress. And I want to ask of you here today to please spare a moment for them if you are in a position to discourage bullies in academia. I hope you will commit to helping less fortunate colleagues in other parts of the world. I want to do the land acknowledgement, and I hope it is not disrespectful that I'm doing this at the end of my talk. I wanted to show my native identity first, and I want to read the land acknowledgement in the traditional pronunciation in the way that Native people call themselves, because it resonates with me, and my pronunciation will not be perfect. I will falter. Please have some grace for me, but I'm doing this from my heart. But by doing this, I am in no way comparing our histories. So I acknowledge that the Field Museum and I am standing here today on the lands of the following sovereign nations: the Anishinaabe, the umbrella term for Potawatomi, Ojibwe, Odawok, as well as the Ho-Chunk, Jiwari, Nutachi, Bakoje, Kiyash Machitiwok, Meshkwa Kia, Aso Kiwaki, Miamiawaki, Wayatanwaki, Piashikaki, Kikapoe, and Anuka. I want to especially thank Kevin Lewandowski and the IT crew for working so hard for me to be able to use my Mac. I am PowerPoint illiterate, and also want to give a special shout out to Women in Science for buying me this brand new computer to support my research grant. And also want to give a shout out. I'm not bad in field work. I don't have a grant, but if you need a field assistant, hey, I'm available. And I also want to give a quick shout out to Cypress Stein Foundation. I am not American, but that doesn't stop me from thanking donors for the recent 1 million grant to the field. And I want to end my talk with another Naga ululation or war cry. In the Field Museum, after I joined here 4 months ago, I have cried a few times at work and at home out of joy and gratitude. I'm so happy to be here as a new member of a big family, and I want to thank all colleagues and friends who have been so wonderfully supporting to make me feel welcome here and for helping me navigate my new life and my new job and life in Chicago. Thank you all so much for coming and for your attention. And I'm happy to take some questions if we have time, but well, Matt Nelson took a bit of my time, right? So sorry with it. And as a joke, so I'm the last spring seminar speaker, and you know the spring peeper, the frogs. Yeah, so I'm like the last spring peeper. I hope you all have a very good summer. If you have some questions and we have some time.
Thank you so much, Chun. That was wonderful. It's about 7 past, but if you have a few minutes to—
Yeah, I have a few minutes for people who can be here.
For those who have questions in the audience, if you can use the mic so people in Zoom land can hear your question, I'll pass it to you.
Hey, Chun. Amazing talk. Yes.
Thank you.
So it seems after you start looking, you found a lot of Sicilians,
Yeah.
like everywhere. Are they generally like this close to surface? Is it only digging, or is there a diversity that is out of reach, like going deeper in the soil, or is it just a matter of people have never looked for them?
Yeah.
They're easy to catch.
Yeah, I mean, very occasionally, if there is lashing rain, you would spot some Sicilians, also because they are disturbed by human beings. They're trying to find new habitat, so you find them crossing the road and all. But otherwise, at least in my experience in Northeast India, there are dedicated burrowers under the soil, and you don't have to dig so deep. Well, men and women have different strengths, but in my strength, and depending on the soil surface as well, like it's just about 40 centimeter. So you dig and you just turn the soil, and if you're lucky, you're there. But sometimes we go weeks without finding any animals. And they can also sometimes be under rotting, like, in fallow land, under decaying organic material or logs. But the best spot is digging, especially near water sources, because they are amphibians, their life is tied to water. So that's the best time to find them, when they are breeding.
One more question. You're going on. Oh, no.
Hi, it was a wonderful talk. Thank you very much for sharing it with us. I feel myself being from South, how it's emotive for you to talk to here today. My question is more about your research. You said that it was a holotype in peace, and then you found more species after you start your PhD. Do you designate an neotype for that, or you just let the holotype the way it was, like now they are together, you have new prototypes for that, or how you handle that?
No, we didn't so they were poorly, they were in very bad states, but we were able to recover most of the taxonomically important characters. So we did not designate new paratypes. The head is broken and all, but we can still take a lot of the measurements, and we can count the scale, the rings, the number of annuli, and also, yeah. But we found a lot of them. It just turns out that no one went to look for them, And the locals knew about them, but they thought they were snakes, and they were just busy killing them.
I have one more question. Good? Great. So when you don't know anything, it's super hard to know where to start, right? So you decided to work for a PhD on this group of organisms that, like I said, they are known for for not knowing anything about them. Where do you get the information on how to go about collecting them, or finding the best sites, or that kind of thing, or did you figure out things that people didn't know? Did you ask for information elsewhere?
Yes.
Just papers?
The only information I had was the few literature, but I had my entire, so I feel attached to my PhD a lot more because it was just not my effort. Like all my family, I mean, Northeast India is so inaccessible, and it only works by having strings of networks, and I have a lot of bureaucrats in my family. So I would call up, hey, uncle number one, I need the state permission. And there's no internet those days. So I had a lot of strengths. So everyone helped, all my family, extended family, circle of friends, and then just find out from literature and try and find out. So the literature, the names and the local names, the type locality in the literature is also you can't find, so you do a lot of interviews. So long story short, yeah, I had to figure out everything myself. And so actually that species that was thought to be extinct, so I called up several uncles to try and find out. I found a spot, I went there, I dug, and in 1 hour they were there. you know? And then learn the trick. So we just go, hey, so our day in the office will start in the fields. We're just going around. We see some place where we can dig or has water. We just do. It's just like hit or miss digging, and yeah, permitting was very difficult, like extremely difficult. But there's a lot of bureaucrats who helped a lot. So all my publication, I have very long list of acknowledgement. People giving me a free Jeep, or the village chairman turning up with a rooster and saying, "Welcome, madam, and thank you for coming to our village to document our biodiversity," and stuff like that. So it was really fulfilling.
Hey, Chun.
Hi.
This talk was so amazing.
Thank
I
you.
am so proud of you. This was so great.
Thank you.
One thing I'm curious about, you mentioned that you started out in academia doing more botany and then moving into the world of caecilians. And like, do you remember when you first learned about caecilians, or like when you first started studying them, you know, even if you'd heard about them before? Like, yeah, tell me about that transition to working with them, because they are such an unusual, understudied animal.
Well, you saw my CV or biodata, right? I had no idea. I just knew that frogs had 4 legs, they had slimy skin, and they go through metamorphosis. I did not know about caecilians, but I knew that I wanted to do a PhD in Northeast India. So when I finally found a professor who could like fund me, supervise me, my first couple of trips were actually on frogs. But I went around with a poster doing interviews to local people and saying, "Have you seen this?" And they say, "Yeah, we know it's an eel, it's a snake," you know. And then we started finding them. And then I got introduced to the British collaborators who taught me all about caecilians. And then I decided, like, okay, I'm really just going to do a PhD, because no one else has done it, and probably no one else will do it, because it's just massive. Like, I mean, it's dangerous, it's expensive, it's a lot. So that's how I started. I had no idea, like it was just figuring out. And it took me nearly 8 years to complete a PhD. And but it was really fulfilling, because there's more. Did I answer your question, Kate?
Yes, absolutely.
Yeah.
Thank you so much, Chun. Thank you all for attending.
Thank you all so much.